Hello everyone, I am Samarpita Gayen from the Department of Biotechnology at the Baba Saheb Ambedkar Marathwada University, Aurangabad, and as part of the HRDC Pre-PhD coursework 2021, today I am going to be presenting on the topic Study of Multi-Drug Resistance Properties of Pseudomonas species. At the outset, I would like to thank Jitendra Kulkarni sir, who is the coordinator of the Department of Biotechnology, for his kind support in this endeavor. Now, I am going to present on the topic study of the multi-drug resistance properties of Pseudomonas. So first we are going to talk about what is the problem statement. So the problem of multi-drug resistance in Pseudomonas species. So as we all know, Pseudomonas is a pathogenic bacteria. It is gram-negative, it is motile, and it causes a nosocomial infection, which is the infection present in hospital settings. It mainly affects the patients who already have an immunocompromised system. So in patients who are suffering from cystic fibrosis or who have burn wounds or patients who are suffering from urinary tract infections, they are the ones who are susceptible to the pathogenic Pseudomonas infection. Now, Pseudomonas is an opportunistic infection, and in most cases it causes a secondary infection which can often turn out to be fatal. So, although there are treatments available for Pseudomonas infection, but in the recent decades it has been seen that there is a high possibility of the development of antibiotic resistance in the Pseudomonas species. So we are going to talk about the emergent multi-drug resistance property of the Pseudomonas and how we are trying to tackle the problem through this study. Now we want to study the emergence of the resistance in the different strains of bacteria, the both resistant and susceptible strains. So what we do is we obtain the samples from the different Pseudomonas species that we have, and we check their resistance and the susceptibility profiles. That is, we identify the susceptible strains and the resistant strains of the Pseudomonas that we obtain. Now, we identify the drugs which are commonly used against the Pseudomonas species, and with them we treat the samples with the drugs and their drug combinations, and then study the characteristics of the bacterial cell. So, we have to compare the characteristics of the bacterial cell before and after treatment, and that is the crucial information which will help us to identify how the antibiotic resistance is occurring. Now, we identify and characterize these differences, and then using that information we suggest the possible different mechanisms through which the drugs can be used to target or a possible mechanism of efficient treatment. Now, the mechanism of action. It is known that antibiotic resistant genes are the ones who are responsible for the development of antibiotic resistance. Now, understanding the difference in this genetic profile can help identify the ones who are the genes who are leading to antibiotic resistance. Now, studying the differences in these gene products can help us identify the possible molecular drug targets to combat the development of resistance. Multi-drug therapy involves the identification of more suitable treatment options for the bacteria in vitro, and different strains obtained from the different environments will definitely have a different susceptibility profile, and this information can help us in producing new targets of the possible drug treatments. And what are the targeted outcomes? It is expected that the knowledge about the why and how of the development of resistance can help us to arrest the development of resistance. Genes from the susceptible strains can be induced into the genes of the resistant strains of the bacteria, and this can help us overcome the development of the resistance profiles. And knowledge of the mechanism and the target of the drugs which are used can be used to synthesize particular targeted synthetic drug delivery systems, which are also used in nanobiotechnology, and this can help us have more targeted approach to the therapy of Pseudomonas infection. And that is all. Thank you.